Okay, we'll get started. And usually Traco is on Monday afternoons, but today, because of the Columbus Day holiday, we're lecturing on Tuesday. So our first speaker is Yves Pommier. He's chief of the Developmental Therapeutics branch. He's a world's expert in topoisomerase. He received his MD and PhD degrees from the University of Paris in France. He's been at NIH for over 3 decades. And he's co-chair of the Drug Discovery Committee at NCI. He's also a member of the Molecular Target Steering Committee. And his title today, DNA topoisomerases and their poisoning by anti-cancer and antibacterial drugs. Yves.
Thank you. Okay, thank you, Terry. So these are 2 reviews that you may want to look at if you want more details. Probably use them as reference. The first one will be detailed, and the second one will be a build-up on the first, giving you some what we have learned from the topoisomerase field, and then the challenges ahead of us. And I'll give you some of that during the lecture. There's a second way you could get access to the topoisomerase world, which is through a book which I edited a couple years ago now, and you could probably get it at the library, and you could go through it and pick and choose what you like. This is focused on cancer. It goes from the etiology of cancer to the treatment of cancer, because topoisomerases are very prevalent as a cause of cancer, as an avoidance to cancer, and as a treatment for cancer. So, and the reason it's not that simple is that humans have 6 genes. So human cells have 6 genes, not counting SPO11, which is for meiotic cells. And in meiosis for recombination, one of the topoisomerases 2, topo2b, SPO11, is used for that purpose. But here we'll just discuss the topoisomerases involved in cells, beyond in somatic cells. And as I said, there are 3 types, and here I put 3 colors, making 6 genes, and in each group there are 2 genes. So on the left side, on the blue, you have the topoisomerases 1, and there are 2 in humans, and in all vertebrates. There is TOP1, which is a gene for the nuclear genome, and there is TOP1MT, which is also encoded in the nuclear genome, but which is for the mitochondrial genome only. So each of our genomes has its specific topoisomerase 1. The second group are the topoisomerases 2. And the topoisomerase 2 are divided in 2 enzymes, the topoisomerase 2 alpha and topoisomerase 2 beta. So in all vertebrates you have, and even Drosophila, you have these two genes. They are larger proteins. You see the molecular weight is indicated at the bottom for each of the monomers. So, I don't know, for each of the monomer. The third group are 2 enzymes as well that are gaining momentum in biology, not in drug discovery yet, but in biology, especially in neuroscience, for topo3 beta, and then for replication for topo3 alpha. So what are these things? So topoisomerases, as their name indicates, changes the topology of DNA, and the DNA being a very long polymer, it is intuitively obvious that when you have 2 meters of DNA in a single cell packed into a single nucleus, there will be knots, there will be entanglement, and especially when DNA is replicated, because you need to make 2 copies. And so the topoisomerases is really what enables the 2 genomes, sister genomes, to segregate and to be orchestrated. So the division of labor is the topoisomerases 1, should really be referred to as a DNA untwisting enzyme or swivelase, and the way they do the job is they break the DNA backbone, and if there is any supercoiling, then it will enable the DNA to rotate. So if you take a cord and it's all twisted, if you cut one of the strands and you let it swivel, it will realign, and then topoisomerase will nick and close, enabling very rapid processing and dissipation of supercoils. Topoisomerase 2 works making a double strand break. So it's easy to remember, 2 for double. Double strand break, and it's a homodimer. and it's a very controlled reaction. We'll get to that later. And then the topo 3 makes single strand breaks, but the difference in topo 3 is they usually work on a single strand piece of nucleic acid. It could be DNA or RNA, single-stranded piece within a duplex. So that creates their specificity. If you go from E. coli to human, because all the topoisomerases are conserved in every organism. It goes from the archaeobacteria to the bacteria to eukaryotes. And in E. coli, for instance, the distribution of labor is different. E. coli has two topo 1A, topo 1 and topo 3. Humans have also two topo 1A, topo 3 alpha and beta, but doesn't have the topo 1. And then on 1B is only on humans. So E. coli doesn't have the 1B, which is the topo 1 mitochondrial. And then on the 2A, on the other hand, E. coli has two pairs, which is gyrase and topo 4. And if you have studied the microbiology and the drugs in microbiology, you should be well aware of gyrase and topoisomerase 4 as targets of quinolones, and a lot of the antibiotics that are used to treat very, very broad spectrum infection, including tuberculosis, which are not targeting gyrase and topo 4, gyrase in tuberculosis. And in human, you have two topoi. So on the therapeutic viewpoint, what has to realize that topo 1 is an anti-cancer target, which is used daily in the oncology field. It's the target of the camptothecin derivatives, and I will expand that. The topo 2s are also the target. The two alpha and beta are the targets of very widely used, almost the mostly used anti-cancer agent, including doxorubicin and etoposide. And topoisomerase 2 alpha and beta are targeted by these drugs. And in the antibacterial field, as I mentioned, gyrase and topo 4 are targeted by the quinolones, ciprofloxacin, a number of quinolones, new generation quinolones, are being targeting these enzymes. So this is a big deal in terms of therapy. How do these enzymes work? All the enzymes have the same mechanism to break the DNA backbone. They work, like, if you wish, like a can opener. So they will open the backbone of the DNA using a tyrosine, and they bring the tyrosine in the vicinity of the phosphodiester bond. And depending on the type of enzyme, it will either form a covalent, it breaks the DNA by forming a covalent linkage to one of the ends. So this is a nucleophilic attack. And in the case of topo 1, the linkage is to 3' end. And for all the other topoisomerases, it's on the 5' end. So in the first figure, you'll see that. The topo 1 is only on the 3' end. All the others are on the 5' end. So in some ways, you could think of the topos as doing reversible translocation, because as they can go forward and make a break, the reverse reaction is that when the DNA is realigned, then the 5' hydroxyl here or the 3' hydroxyl there can attack back. And actually, it's much faster going backwards than forwards. So in your cells, in my cells, these intermediates are almost invisible. We call them cleavage complexes. They're invisible unless you have a drug or damaged DNA. So let's go on to topo 1. So topo 1 was the first discovered enzyme. That's why it's called 1. Actually, the numbering goes historically. It was discovered by Jim Wang in E. coli, and by Jim Champoux and Jules Bécaud for the vertebrates. It was called initially the DNA untwisting enzyme, which I think is the appropriate term. And it was discovered by, once the DNA was discovered, you had ethidium bromide, and then you could visualize DNA. And when one could visualize supercoiled versus relaxed DNA, it became obvious there was an activity in cell extract that could actually magically relax supercoil. And this activity was purified at topoisomerase 1. So what you have, you start with supercoiled DNA. Topo 1 makes a cleavage complex. By breaking one of the strands, it enables the free rotation of the DNA around. And then by base stacking, it realigns, and then it religates. Topo is gone. And this is a controlled rotation. Topo 1 will break. You see it makes a covalent linkage to one end, let the other swivel. but this is regulated. Actually, the speed of rotation, we did this in this building with Keir Newman, is actually very controlled. It's like a magical clock. It doesn't go with the torque. It's actually very, very independent, somewhat independent of the torque. So it's a very beautiful little machine. It works without magnesium, without ATP, doesn't consume any source of energy, and even works at 0 degrees. So you could relax DNA at 0 degrees by extracts because topoisomerase 1 is active. It is essential for transcription and replication, and if you knock it out in mice, it is early embryonic lethal. It is embryonic lethal in Drosophila, but not in yeast. So yeast manages to use the other topoisomerases to complement, but not the more complex organism. So if you think of it, so let's say you have DNA, DNA is being opened up to be transcribed or replicated, and as you do this intuitively, you realize you're going to build supercoil, what we call supercoil, like a rope, separate two strands, and if Terry is holding one hand, you'll see as I do this, you'll get these bundles. These are supercoils, and if there are too many bundles, you can't separate anymore, so topoisomerases are coupled with transcription, replication, and chromatin remodeling to enable the dissipation of these supercoils. So ahead of these, you have positive supercoil, and behind you have negative supercoil, and topoisomerase 1 will dissipate these supercoils. It does it very well, very fast, about 6,000 RPM on normal DNA. But if the DNA is somewhat damaged, it can get stuck, and you get these cleavage complexes that can happen if you have damaged DNA or if you have a drug in the cell. And I'll show you how the drugs work. So bear in mind that in addition to drugs, in fact, top-1 cleavage complexes are induced endogenously and also during carcinogenic exposures, abasic sites, carcinogenic adducts. And every cell has a machinery to repair this lesion. I will allude to that. So this is built in the system. There is a rescue mechanism when these topoisomerases get stuck. So the two human topo1, so this is a scheme, you just keep it for your slide. This is the nuclear topoisomerase 1. This is the mitochondrial topoisomerase 1, and this is an alignment of their structure. They are both coded in the nucleus, as I said, and we discovered the top1MT in 2000. That was the last topoisomerase discovered in human cells, I think. So that's the 6th, and that's the mitochondrial topo1. It has a mitochondrial targeting sequence in the N-terminus. And these are the topoisomerases for more primitive organisms, so such as viruses. Vaccinia virus encodes its own topoisomerase because it replicates in the cytoplasm and needs to have its own topoisomerase. But you have also some bacteria. Some of them have a topo1b, and then the structure is very conserved, but usually doesn't have an N-terminus domain. And all of these things work by the same way. They relax supercoil by swiveling. So it's just this rotation swiveling reaction. They are targeted. In about year 2000, 2 camptothecin derivatives were approved by the FDA and now are used in the daily oncology. The two that are approved are irinotecan and topotecan. And the reason they came about is that camptothecin is very insoluble. And then the drug companies decided to make water-soluble derivatives. And if you look at the structure, and you could look at your leisure later, but this is the structure of camptothecin, all was added were these red residues, and these red residues confer water solubility. In the case of irinotecan, this water solubilizing group, though, inactivates the drug, and it needs to be activated to the active form. So when you say irinotecan as a topo1 inhibitor, it's not a direct, it's his metabolite, which is bicarbosylextrase, SN38, which is active. But in the clinic, that's what it is. One thing you have to bear in mind that we knew from the beginning when I started working on the camptothecin before they were approved is camptothecin is a natural product. So it's from the Camptotheca cuminata, a Chinese tree, was discovered by the NCI as an anti-cancer agent. We immediately, and Monroe Wall, who discovered camptothecin and Taxol with Mansukh Wani, immediately realized that this is actually quite unstable. Normally, under normal conditions, camptothecin has rings open and this is inactive. So when patients are given either topotecan or irinotecan, oncologists today tend to forget that these drugs are very unstable, and so they take for granted that what they give is what the patient gets. Actually, the patient gets only a very small fraction of the active form. So there's an intrinsic problem with these drugs. What we have learned with the camptothecin was actually quite remarkable. For many years, many people had tried to understand how the camptothecin were killing cancer cells. They were seeing the breaks. They couldn't find the target. But even when the target was discovered as being topoisomerase 1, it was found that it was an absolutely selective target, because in yeast, for example, yeast could be killed with camptothecin because it has topo1. But if you knock out topo1, yeast is totally immune. It means that the only target of camptothecin is topo1, because if you take it away, yeast is totally insensitive. And it's a paradox, because why, by having the target, are you killing the cell? Well, the reason you're killing is that normally topo1 cleaves and religate all the time. And under normal conditions, the religation is more efficient than the cleavage. So you never see this normally. And this is pretty dangerous because you're making a single strand break with topo1 at the 3' end. What camptothecin does, and that's what we postulated actually in the 90s, is that camptothecin binds at the interface of the topo1 DNA complex. So this was proposed at that time, and it took about 10 years to confirm by co-crystallographic studies that this is indeed the case. So this is the DNA in green, the break is here, the topo1 is in gray, and you see here the topotecan is bound exactly like I drew here. And if you look at the surface representation, it's amazing how topotecan find its way into this cleavage complex and bind just at this interface. So that taught us a lesson which was not obvious in pharmacology, because everybody was thinking of competitive inhibitor, allosteric inhibitors, and things of that kind. This is another kind. Actually, the drug binds the interface of two macromolecules, the DNA and the protein. It doesn't bind to DNA alone, doesn't bind to protein alone. It only binds to the complex when the complex has this little cavity, which is the break site. And nature has found a way to select this to make a toxin. We use it for cancer treatment, but that's really the way it was. And that led to this paradigm of interfacial inhibitor. This was, so this is the same if you wish. In fact, in the 90s, when camptothecin went into really the clinical development, and knowing their shortcoming with the chemical instability, we decided to look for new topo1 inhibitors that would not have this chemical problem. And as we're doing this, we found them, and we got co-crystal structure. And this is one of these drugs. You could see it sort of looks like a camptothecin, but it doesn't have this chemically unstable ring. And again, this DNA is in blue here, the topoisomerase is in yellow, and the break is here. And you could see the drug, it's stacked between the base pairs on the cleavage side. It's like a sandwich. So it's like the slice of cheese or ham, and then you have the two pieces of bread, which are the base pairs, and it's open. And it only binds when this is open enough that the drug fits in, and this is the catalytic tyrosine that covalently attaches the topo to the end of the 3' end of DNA. So again, this interfacial inhibitory principle extended to this type of drugs. And actually, over time, we've expanded this. All the topo1 inhibitors work that way. They all have this interfacial type of process. But what you have to realize is a lot more interesting than just the drug stacking like we represented here. This is the cleavage seen from the side, where you have the base pair. So you have one base pair here, one base pair there. This is the 5' end. This is a 3' covalently linked. This is the drug. You can see how nicely it fits, and you turn 90 degrees. It's like a really nice sandwich, not very much getting outside of the bread. So it's pretty tight. But what's remarkable is the drug not only binds DNA, but makes a network of hydrogen bond to the topo1 itself. So it's anchored by pi stacking and by hydrogen bond. And if you mutate any of these amino acid residue, you confer very high resistance to the drug. So everything has to come together for the drug to work. Therefore, it's really ternary complex. And in fact, if you look at the camptothecin producing plants, Because camptothecin is produced by nature. So you would wonder, how do the plants survive the fact that they produce a toxin? And remarkably, what was found is for many, many of these plants, they have a mutation on the TOPO1 gene of the plant, which is equivalent to this one. And by mutating that to asparagine to serine, the plants are immune to the toxin they produce. And in fact, what was so beautiful is that when this was discovered in 2008 and published, I got an email before the publication saying, please open the attachment, and which I did because I'm at NIH, so I thought the computer people could rescue me if I did something wrong, and also because it seemed to come from Japan, it looked reliable. And what I found is actually, the reason they sent it to me is that several years before, we had actually selected a human cancer cell line resistant for camptothecin. They had exactly the same mutation. To me, that's quite remarkable. It tells you these drugs are very, very highly targeted, but it tells you the story how nature manages to make these toxins. So we developed new TOPO1 inhibitors. I alluded to you, and the reason I'm talking to you is this drug is just not a lab exercise. As I'm speaking, these drugs are used in the clinical center here. They're in phase 1 on the clinical center, and they are throughout the United States in an NCI-sponsored, what we call the Comparative Oncology Program, where they are tested in dogs in veterinary clinics. People bring their dogs the dogs have primary lymphoma, and then they're treated with this drug, and then we do the clinical trials. And they're active, actually, in the dogs as well. So it's actually rewarding in the end. So the reason we did this is we knew camptothecin were effective, and we also knew that within each class of target, if you take different chemicals, they usually have different clinical spectrum. So that was a no-brainer to do that. And in the TOPO1 field, for some interesting reason, the camptothecins are the only chemical class targeting TOPO1. And I think the reason is that historically, they were the last chemotherapeutic cytotoxic drugs to be marketed before the age of what we call targeted therapy, which were all the IBs and all the kinase inhibitors. Now it's coming around because you get the PARP inhibitors coming back, so you're getting back into chemotherapy. But there was a gap of about, I would say, 15 years. So camptothecins were the last one. Nevertheless, during these 15 years, we have been developing our own, and they overcome the limitations of camptothecin. They are less bone marrow toxic. They are not intestinal toxic. The irinotecan is pretty toxic on the gut. It was almost abandoned clinically in the early clinical trial because some patients have so severe diarrhea. But some smart MDs were able to overcome it, so the drug made it to approval. And they also are not drug efflux substrate, and our drugs are chemically stable. So this is what I meant before. In camptothecin, this is the active form. In the blood, within minutes, camptothecin is converted to this, which binds serum albumin, and that's a problem. So these drugs were discovered from the ones I'm talking about, the non-camptothecin, were discovered when I looked with my colleague at the Developmental Therapeutics Program at the collection of drugs in the NCI database. You may know or may not know, but the NCI is a very large repository of drugs, about 500,000, that have been characterized through 60 cancer cell lines. And when we looked, and I looked for drugs that had the same profile of cytotoxicity activity, and I don't really want to go into detail, but this is a cytotoxicity profile across the 60 cell line. And each cell line is either more resistant or more sensitive. And using this, then all we did is look in the collection, if anything looked like this, but was different chemically, and that came back with this. So when I found this in the, I think it was mid-90s, it had been put in the database by a chemist from Purdue University, Mark Cushman, and Mark had put this in the database because it was something he had made in his synthesis and didn't know how good and for what it was. So it sat in the NCI database for several years before we picked it up, and then I called Mark, and after that we've made several hundreds of drugs, and these are the ones that are now in the clinic. So these are just all these compounds, and the nomenclature I should have updated. Initially, they had this really code name. Now they are referred to as LMP744, LMP because that's the name of our lab, I guess. and the 3 digits, LMP744, LMP776, and the last one is LMP400. So these are the 3 that are in clinical trial. And so when we put them in clinical trial, what happens now is we decided to do pharmacodynamic biomarkers to follow the activity of the drug, because you have to realize that nowadays, with a cytotoxic, it's very difficult to put it in a clinical trial without having a biomarker. You would want to know quickly in the patient whether the drug is really doing something to the tumor, and you don't want to wait for eventual responses. So we developed 2 biomarkers at the NCI. One was the gamma H2X, and still is the gamma H2X. The other one is topo1 itself, which is the target. And to do this, we teamed up with the NCI team in Frederick and with Jim Dorosho and all his pharmacodynamic biomarker team, and developed these 2 biomarkers. So the gamma H2AX, you may know, I think most people in the world today know gamma H2AX because it's one of the first DNA damage foci biomarker that had been discovered, and because many people publish on DNA damage using gamma H2AX as a chromatin marker of DNA damage. You may know or may not know, but this was discovered here. Bill Bonner, William Bonner, he's a co-worker, he's in our lab, and Bill Bonner discovered gamma H2AX. Called it gamma because he discovered with gamma radiation. So with Jim Dorosho, who is the director for translation research at the NCI, and Bill, we decided to do this biomarker. To do it, it's easy to say, but it's much harder to do. We had to go back to mice and show that this gamma H2AX biomarker, which is chromatin damage, would respond to an already approved drug. So we went backwards and took mice, treated with topotecan, various doses, and seen whether this biomarker would work. That would establish whether you could see this at pharmacological dose, and at what time you would do it if you were to biopsy patient. You cannot biopsy patient over and over. You have to make a decision. Is it 12 hours after the infusion? Is it 6 hours? Is it 48 hours? So the mice could help us. We did that. So this is to show you what it looks like. These are 4 different tumors, and gamma H2AX positive cells, nuclei, are green. So this is the untreated. These are the treated. So you could see little green things. So these are the cells that have been damaged. And if you expand, you see that here. And this is with our non-camptothecin topo1 inhibitor. We see it with topotecan. We could see it with the LMP drugs. This is LMP400. So it works in mice, and therefore we were ready to go to clinical trial. And as we were doing this, the LMP400 shown here had a quite interesting, nice anti-tumor response. So this is the tumor growth in the absence of drug. This is the tumor response to topotecan at the maximum dose, and the mice at this dose are really sick because it's toxic. And with our drug, we could do better with less toxicity, and we get gamma H2AX. So we went on, and that's why it's in phase 1. And the time, turns out that you could do the gamma H2AX probably at 4 hours after the infusion, which would work clinically if you treat the patient in the morning. You could do biopsy in the afternoon, or 7 hours, which is harder because then that would really put you in the night. The second one we did is topo1, so this was also validated. That led to an ELISA assay, and in this case, what happens when you treat with the topo1 inhibitor, you have the engagement of topo1 being poisoned by the drug, and then topo1 goes down. The cells manage to respond by putting topo1 down, so it's second pharmacodynamic biomarker, and these have been integrated into the clinical trial, and they will be deconvoluted once the clinical trial ends, which hopefully will be I think early next year, to be realistic. So let's talk about Topo2, unless you want to take some question on Topo1. It's up to you. If you have something you want to ask, you're welcome to do now. Okay? So we'll get to the second pill now. Topo2. I'm not going to say anything about Topo3, so that's the good news, but it's unfortunate because it's very interesting, but it's really biology at this stage. Topo3 is a fascinating biologic target, as I said, for neuron, for DNA replication in conjunction with Bloom helicase, and for neurons with some RNA binding protein. But there is no drug, and I don't have enough time. But we're writing a review, so hopefully when the review comes out, we'll put the 3 topos in there, and then we could read them. Topo 2. So what is the difference between Topo 1 and Topo 2? I've alluded to the fact that Topo, well, beside the fact one is blue and one is red, but that's not what I mean to go to. You could already capture some of the differences. The DNA is always in green. The DNA is being worked upon by breaking the backbone. That's common. But what's different? Here you have only one ball. So Topo 1 works as a monomer. Topo 2 works as a dimer, 2 balls. And usually each ball takes care of one strand, but actually they're really coming together. This is linked here. Topo 1 doesn't use ATP. I've mentioned that. Topo 2 needs energy. It burns ATP. It requires magnesium. So it's a more complex enzyme. It doesn't work at 0 degrees. Topo 1 does. And the drugs are different. There is no overlap between a Topo 1 and a Topo 2 inhibitor, which is quite remarkable. And there is no overlap between a Topo 2 inhibitor in bacteria, the quinolones, and the Topo 2 inhibitors for cancers, etoposide doxorubicin. So you may know, but one of the person who discovered topoisomerase 2 in bacteria is here, is at NIH, is Marty Gellert. So Marty Gellert discovered gyrase, and when Marty had gyrase and I was working with Topo 2, Marty gave me when he knew I was working, gave me the quinolones. And I checked, the quinolones don't work on the human Topo 2. So it's really specific, and you'll see why. So, and then the difference is Topo 1 links to the 3' end. And that is quite peculiar because the 5' links to the 3' end actually puts Topo 1 in a class of enzyme like the recombinase. Lambda integrase is a 3' tyrosine linkage protein. CRE, which you use for all your recombination reaction, is working that way. And you may have bought the kit, the Topo kit, to do your recombination reaction. It uses Vaccinia Topo 1 because it has this property to religate very well. So this is a particular type of topoisomerase with potential danger of recombination. But Topo 2 is linked to the 5' end. As I said, it's a dimer, and the drugs are different. So why 2 Topo 2? What are the differences between the 2 Topo 2s? So if you take the Topo 2 alpha versus the Topo 2 beta, the main difference is in the C-terminus region, in fact. And the C-terminus region appears to be the one that interacts with other proteins, including chromatin remodeling complex. And also, it has a difference in the way it relaxes positive versus negative supercoils. The Topo 2 alpha is essentially expressed in highly proliferative cells and usually coupled with DNA decatenation during replication. Topo 2 beta, on the other hand, is expressed in every cell, including neurons. Neurons do not express Topo 2 alpha. They do not replicate. Cardiomyocytes do not express Topo 2 alpha. They do not replicate. But they have a lot of Topo 2 beta because they transcribe. So Topo 2 beta is a cofactor, a necessary cofactor for transcription, probably breaking promoters, and things that are still to be totally worked on. And then if you look at go to the bacteria, there is a very high similarity in the way these things are constructed. But bacteria use actually, instead of one protein in gyrase, uses gyrA and gyrB. So you have 2, and then they come together, okay? And the reason they use magnesium, there's beautiful biochemistry, which is conserved through the bacteria and the human enzymes. And it just is a way to position the DNA with the enzymes and coordinate everything. So the reactions, so Topo 2 catalyzes a broad range of reaction. And the reaction of Topo 2 operate like the gates of the NIH. So if you could remember the way that works, it's a 2-gate mechanism. So imagine that you want to pass one strand through the other. So there is a gate strand or the transport strand. So you want to get one through the other. Well, you could do it really by totally opening and let go, but it's not the way it works. I said it's like the NIH gate. The strand that needs to pass through the other first engages on the top. Then the top closes, like you go in the NIH entrance, you're in the middle, and then it opens the second, and then gets out, and then tredigates. This is a very controlled mechanism. All the time, the topoisomerase 2 keeps the ends together. They don't come apart, because if they did, it would be extremely dangerous. You'll just break the DNA. So it's a two-gate mechanism. All the topo2s do that. And by doing this, they could do miracle things. If you take two DNA circles that are like this, it will decatenate. It could go the reverse, it will catenate. It'll take a knot out, same way. It'll just pass a strand through the other within a molecule. It will unknot, and it will relax, because it could still do that. And it will induce negative supercoiling in the case of gyrase. So I just indicated here what is common to each. So you could see decatenation and unknotting is common to all the topo2s, and the relaxation of supercoiling is common to the topo2 and the topo1, and gyrase is the only topoisomerase that can induce negative supercoiling in bacteria. This is a scheme of the drugs, and the point of this is to show you that by contrast to topo1, where you had only one chemical family, the camptothecin, in the case of the topo2 inhibitors, there are a number of chemical families. And the three main families are the etoposide family, which from natural source initially, dimethyl-epipodophylotoxins. They have a sugar, and then they have this portion, and that is appended ring. The second big family is anthracycline with doxorubicin. These are planar molecules with, again, a sugar here. And then the third are the mitoxantrone, which is used for cancer or some immune disease. And these are some experimental drugs. These are the antibiotics. You could see they don't really look alike, yet they will have the same mechanism of action. By mechanism, I mean interfacial inhibition. Once again, it's the same mechanism as topo1. This is remarkable because the enzymes are different. They break the backbone. They have different structure, yet the way the drugs work is the same. So if you look, for example, as a topo2, so this is a crystal structure of topo2. The DNA is barely visible. It's in the middle. And here you just flip it 90 degrees to see the top. So now the DNA is lying on the top. You could see it. And when you have the enzyme is poisoned by a toposide, the drug is exactly in the cleavage site. So you can see how beautiful the drug actually snuggles in the cleavage site, and again makes DNA and protein interaction. They're highly specific. If you mutate one amino acid residue, the enzyme becomes totally resistant. So it's a fit. And this is, again, the principle of the interfacial inhibition. And then you find that the bacterial drug do the same. If you take now the structure of topo4, and there are several of these structures nowadays, and this is fairly recent. I'm not talking things that were done 20 years ago. This is within the five years. In gyrase, and this is quite remarkable, if you look at, this is topo2 human, this is gyrase bacteria. They're not so different when you look. But the thing is the same. The drugs bind exactly in the cleavage site. But because the enzymes are different and the hydrogen bonds are different, the drugs do not overlap between gyrase and human. So they're highly specific because of this protein drug interaction. So these are the antibacterial. And as I said, so that led to this idea that initially was a while ago that we first, there was 2005 when first coined the term of interfacial inhibition. And I must tell you, this is not limited to DNA-protein interaction. And the reason we have two authors here is Jacqueline Cherfis had actually shown the structure of briferdinate in protein-protein complex, which was the ORF complex. And it's the same principle. Protein needs to move around. And as they move around, they create little cavities, and natural toxins fall in the middle and blocks them. And that's very toxic for the cell. So that's the notion of interfacial inhibitors. We expanded this a couple of years ago, 3 years ago, if you want to read about this. There is a more recent one that came about. Because I think for drug discovery and for general biology, this is quite remarkable. It's not something that is totally on the surface of what people know or are being taught. So to finish, I'll give you a little flavor of the fact that with this enzyme, the topoisomerases, come a danger, which is that not only when you put a drug, but topoisomerases tend to get stuck on DNA, and cells are equipped to remove these protein DNA complexes, and they have their own machinery. Well, I'll tell you one little thing that's kind of interesting is if you compare, so you may wonder, well, let's go this way. So maybe it's easier. So what is the problem? The problem is when you make a topoisomerase cleavage complex, so if it's topoisomerase 1 cleavage complex, I've indicated to you that the topoisomerase gets stuck on the 3' end of DNA, and this is a covalent bond between the tyrosine and the topoisomerase 1. This is covalent. This is not going to break, so you need to remove that protein actively. And in the case of topo 2, the covalent bond, and topo 3, the covalent bond is at the 5' end. And again, this is a tyrosyl bond. This is not going to break by itself. You need to go at it enzymatically. And cells have developed specific enzymes to remove this, which are called, because of the tyrosine, tyrosyl DNA phosphodiesterase. So the first tyrosyl DNA phosphodiesterase was discovered here at NIH by Howard Nash, who had worked with Marty Gellert and Kiyoshi Mitsuushi for the discovery of gyrase. And Howard Nash was working on lambda integrase and was always curious of how could cells eliminate, repair this, and discovered TDP1. So TDP1 breaks this and can free the topoisomerase, regenerating the end. Then a few years ago, a second TDP was discovered for the topo 2, which was then called TDP2, and it removes that end. But cells actually have more than one way, because these lesions are so detrimental potentially that they also use an alternative pathway. And we know this from genetics. Using the drugs, again, you could see which cells are particularly sensitive. So I'll show you an example. So this is for camptothecin here. This topo 1, topo 2, and topocyte. And this is a bunch of DNA repair cell line. So this is a list. So this was published actually a year ago. But if you look for which pathways are particularly determinant or sensitized to the drug, so for topo 1, no problem, is TDP1, okay? But it's not only TDP1. And for topo 2, this is TDP2. But look, this is what we knew, but there's a lot more. So for topo 1, all the HR genes actually are involved. The Fanconi genes also are involved. So it means the cells are really not using just one thing to remove these. And what we know now is a bunch of endonucleases that can actually cut the piece of DNA to which the topoisomerase is attached, and then the cells can then fill the gap and repair. So there is a balance between all these pathways. And in the case of topo 1, I've mentioned that. So why is the topo 1 cleavage complex toxic? The toxicity of a topo 1 cleavage complex in the presence of a drug is toxic because normally this is reversible. Even when you have a drug, the drug binds and comes, and nothing much happens if the cells do not replicate. But when you have replication coming in, because the topoisomerase is linked to the 3' end, just the mechanics of the system, is the DNA polymerase then collides into this topoisomerase and generate a double-strand end. And that double-strand end not only generates a topo 1 DNA complex, but needs to be repaired, and you need homologous recombination for that. And that's drawn this sort of what I drew it already. TDP1 removing just the topo, tyrosyl DNA phosphodiesterase, and then all these endonucleases, and some are really very prevalent in biology, XPF, ERCC1, MOSET1, EME1, MR11, RAD50, NBS1, CTIP, all of these are very prevalent in DNA repair nowadays. All of these are involved in this repair reaction, and many of these are actually mutated in cancers. So the other thing that is emerged about last year is actually this repair enzyme, TDP1, doesn't work alone. It has a cofactor, and this cofactor is PARP. And PARP inhibitors actually PARP drives TDP1 to repair. And if you use a PARP inhibitor, de facto, you block TDP1. Therapeutically, this has an implication. because let's say if you have a cancer which is deficient in ERCC1, and that happens, then if you block TDP1 or if you block PARP, then you have synthetic lethality. So there's a testable implication. The combination of a PARP inhibitor with a topo-1 targeted drug, which we know is very synergistic, should be selective for the tumor if the tumor is deficient in the endonuclease pathway. So if you have an XPF ERCC1 or MR11 deficient tumor, colon cancer, then it's not unlikely you'll get selectivity towards the tumor. And the clinical trial then could be designed based on this synthetic lethality. So repair of topo2, I've already mentioned TDP2, so that's the other side. And in this case, what the coupling of the TDP2 repair works is, okay, you have topo2 cleavage complex. If it aborts, you get the covalent complex, you're stuck. And then the cells usually use proteasome and proteolysis or denaturation, denature the topo, and then the TDP2 can then cleave off, generating a 5' phosphate, and then the next step goes through just the unjoining reaction. So this was published now, and that's pretty clear, and there is a crystal structure of TDP2. So I'd be happy to take some questions. This is just the list of the people, but I think all the work we've done over the years, we've been very fortunate because of the NIH and the NCI environment to have all this incredible connection of people who have worked together to contribute to all this knowledge and work and drugs, I guess. Thank you.
So are the topoisomerase inhibitors effective in all cancers? Or one cancer relative?
No, they're selective based on empirical experience in the clinical practice, and I cannot tell you exactly whether we could actually totally predict it, but if you take the topo1, so topotecan is approved for lung cancer at relapse, small cell. Topotecan uses ovarian cancer, again, in the relapse setting. Irinotecan is widely used in colon cancer in the FOLFIRI combination, so it's quite works okay, I think. For it's a good first approach. So it's different because one is in colon, the other one is in other diseases. And then the topo2 inhibitor, if you take doxorubicin, is very prevalent in many chemos. In breast cancer, for instance, it's almost a must, and the patient will go through doxorubicin. Etoposide has a different spectrum again. So really indicates that our new topo1 inhibitors that are finishing phase 1 is very likely they will not have the same clinical profile. But the challenge for us is, could we help them do that? To find out ahead of time for the phase 2. I have to stop that. I don't know how to do it. Yes, so the profile is clearly very different for each of these drugs. And I think the challenge is, could you anticipate, even within if you take even breast, I mean, is triple negative a good choice for a topo1 inhibitor? I think all the year it's just amazing because, drugs they're approved in the early 2000s, and then the breast cancer community just didn't use the topo1 inhibitor, kept using doxorubicin topo2 inhibitor. And now what I'm seeing is because there are camptothecin that are formulated with pegylation and so on, they're coming back. And triple negative are starting to be treated with topo1 inhibitor. And I'm hopeful that the other topo1 then will have a second life, and we understand better all the repair defects in triple negative or homologous recombination defects. So I think if the tumor is homologous recombination deficient, it would be a reasonable indication for topo1 inhibitor. You okay?
Okay, thank you very much.
You're very welcome. I can do this. Thank you. Thanks, David.
Okay, in terms of announcements, we've assigned the core visits and the visits to the tumor boards, so they will occur in late October, early November. And our next speaker is Frank Madorelli. He got his PhD from City University of New York, his MD from Mount Sinai School of Medicine. He did a residency at Columbian Presbyterian Hospital in New York, and then he joined NIAID. And subsequently, we recruited him to NCI, and he's an investigator in the host virus interaction branch, HIV dynamics and replication program. He's head of the clinical retrovirology section. He's going to talk to us today about retroviruses. Frank.
Thank you very much. Well, thanks a lot, and thanks for the opportunity. And as I was coming over here, I saw that they're reopening Rockville Pike. So I hope these sad looks on everybody's faces isn't that they're going to be this lecture. But I'm actually very excited about talking about retroviruses in this context. And one of the reasons is, as I think you get a sense from Eve, that a substantial amount of this work was done here at NIH. And I think a lot of work with retroviruses, and I'm sure you've heard at least about some of it, has been done over the past 25 to 30 years at NIH studying HIV. But I wonder, if we go back a good bit further, that a great deal of work was done on retroviruses long before HIV was even discovered. So you all are here on the NIH campus. How many of you work in Building 10? Almost all. And so the rest of you are at the outbuildings of the big plantation, right? Which buildings are you in? 35? 35. So 37 was where Bob Gallo used to work. How about you guys here? 50. Anybody in Building 7? Or 4 or 5? Well, 4 or 5 are still standing, but as you leave today, you can see where Building 7 is. And in that building, a great deal of work was done on retroviruses of mice by Wally Rowe and his group, and Malcolm Martin, who's actually still here in Building 5. So this is an NIH deal. It's not just HIV, Bob Gallo and Dr. Fauci. It's a great deal of work that's been done. And a lot of it was done in NCI, and the reason for that is because before these diseases caused immunodeficiency, they caused cancer in animals, and they were wonderful models to study cancer. So we know a great deal about it because of that, and as you know, many of at least one of our former directors, Harold Varmus, got his Nobel Prize studying. So what I wanted then to talk about was to introduce the topic and then to talk about the molecular biology and the replication of retroviruses, because that replication program influences pathogenesis both for the development of cancers in animals and people and for the development of immunodeficiencies in humans. Talk a little bit about these retrovirus infections in human populations, some of the things we've learned about how a virus infection can emerge and spread, and then overall some lessons. So for HIV, this is the overall situation in 2012. And I'm just curious, are these numbers a surprise to you or not? That there are about 35 million people infected with HIV worldwide. How many people have cancer? Yeah. Is so low. I guess it's not included. I'm not sure it's not included with the 1.4 million. That's going to be the United States, which is somewhere around 0.8, 0.9, and the rest in Canada. So I think the 240,000 is the islands, and the 2 million is Mexico and further south. How many people have cancer, Terry, in the United States? So we probably have about 0.8 million with HIV infection and a couple of million at least with cancer. So between the two of them, a substantial disease burden in the United States. Who gets it depends upon a number of factors, as I'm sure you're well aware. But across the world, it changes quite a bit. If you look in Central Europe, former Soviet Union countries, IVDU is about almost 2/3 of all the infections. There was an explosive epidemic in the late 1990s and early 2000s. In Southeast Asia and South Asia, a great deal of commercial sex work is responsible for the numbers of cases present. And here and these are data for the Caribbean, but it illustrates an important point that it can be well distributed among intravenous drug use, men who have sex with men, the typical risk group for the early HIV epidemic, but now not so typical. Commercial sex work, and then there's this other category which we call other. In other words, people are still not self-identifying what their risk is for HIV infection, and the reason is because along with an epidemic of virus infection, there's an epidemic of stigma, which persists to this day. So I think something that you're quite familiar with, molecular biologic techniques, to understand the replication of this virus. And I think some of the things we've learned about HIV are things that you probably use all the time. So if you work in a lab and ever do cDNA synthesis or things of that nature, you've used retroviruses and their enzymes. But in their broadest consideration, they're a group of RNA viruses that replicate by a DNA intermediate using reverse transcriptase. And I used an old font for this to drive home the point that these viruses and their mode of replication were identified not all that long if you take a long view, wasn't all that long after Watson and Crick discovered the structure of DNA and how DNA replicated, and this concept of DNA to RNA to protein, which was the original way things went, right? We start out with DNA and genes, and they make RNA, and then we make proteins. We finally got it right once we figured out what the genetic material actually was. And it wasn't that long after that people realized that there were going to be viruses present that took RNA, and to make more of that virus, you had to go back to DNA, then to RNA, then to protein. And so it's called a reverse transcriptase because it takes RNA into DNA. So quite a tour de force. This enzyme, reverse transcriptase, is then central not only to retroviral replication, but it's pretty important for the rest of us as well. And so this was really a different paradigm for replication. And it probably emerged from the transition from an RNA world to a DNA world. Now, the term RNA world, have you guys heard that before? Is that something that's totally... The earliest catalytic molecules were likely RNA molecules. And so one theory about how life emerged on this particular planet was that it started with RNA, and these ribozymes were able to self-replicate, which worked out really well until some of the byproducts of that process started to accumulate in the atmosphere. And one of the byproducts was molecular oxygen and oxygen radicals. And I don't know how many of you worked with RNA in the laboratory. Anybody work with RNA? Anybody really careful with this particular RNA? Because it degrades so quickly, and you got to buy special water, right? and use RNase away and be very. I'm driving this home only because we didn't have that back when life started, and you know, he's not old enough, but I'm almost old enough to remember that. It was about, 3.8 billion years ago. Back then, there wasn't anything like that. So if you happen to be on this planet and accumulating molecular oxygen in the atmosphere and trying to store the only thing that's keeping us alive, the genetic material in RNA, that's not a long-term plan. And so the organisms that survived, or at least this is the current theory, the organisms that survived were the ones that could convert their genetic material into something that was a little bit more stable than RNA with these crazy vicinal hydroxyls. And that was deoxyribose nucleic acid. And the enzyme that did that was a host reverse transcriptase. And so there's a part of this theory is that reverse transcriptase saved the planet. So probably worth knowing about. The truth is, wherever you go, whatever phyla you investigate, every one of them has a reverse transcriptase-like enzyme in the genome, perhaps a remnant of when RT was saving life on Earth. And so this is just, if you took a walk in the woods, as if you're allowed out of the campus. But if you did, anything that you saw has a reverse transcriptase. Now, if we look at the viruses or retroviruses that also have that reverse transcriptase and classify it by that enzyme, since it's common to all of them, we'll find about 7 major families of viruses that have reverse transcriptase, and they're depicted here in this phylogenetic diagram. And the thing I wanted to point out is that they infect mostly vertebrates, some fish, few reptiles. That's pretty much where retroviruses now. There are retrotransposons and things like that that are everywhere. You can find them as far down the tree as flies, et cetera. But let's just think about these viruses for a moment. And the other point I wanted to raise, or at least remind you, is that the ones that cause disease, at least in humans, lentiviruses, they're all in bold. Lentiviruses, and those are the ones that we know an awful lot about, HIV-1 and HIV-2. Human T-cell leukemia virus, maybe somebody of you are involved with that virus. And spumavirus is largely a virus of animals and primates, but can infect humans. They're not all closely related to each other. They're really 3 independent kinds of viruses that happen to infect humans. So, for instance, these lentiviruses, HIV-1 and 2, they have close relatives, but they infect monkeys, simian immunodeficiency viruses, horses, goats, and sheep. Then this other group of HTLV that causes cancer in humans also infects cows. Spumaviruses mostly in primates and not much else. So just because it infects a human being doesn't mean it's closely related to each other. In the broad scheme of things, yeah, they're all retroviruses, but they're very, very distinct. Here, if we now cone down and look just at the lentivirus relationships, we see a similar phenomenon. So here's HIV-1, he said. Here's HIV-1. Its closest relative is a virus that's in chimpanzees. HIV-2, which also infects humans, it's more geographically restricted to West Africa. It also has a relative in the animal kingdom, and in this case, it's in monkeys and it's in sooty mangabeys. So once again, just because this and this are in human beings doesn't mean they're most closely related to each other. And that observation helps identify where the viruses came from and gives us some sense as to how they spread, how they jumped into the human population. And we'll come back to this. But immediately what you see is, well, maybe this HIV epidemic is really not just one epidemic. Maybe it's multiple epidemics. Just to talk about the viruses and get into the molecular biology a little bit more, there are certain conventions. that if you look at the virus itself, so this is what's in the virion, they're RNA molecules, and they have regulatory regions and coding regions. And I didn't make a mistake here. Strangely, the poly A sequence is all the way at the 5' end, whereas the promoter is at the 3' end. And we'll see why in a moment. Just by conventions, the name of genes are in lowercase. As you'll see, protein gene products are capitalized. So as you read through papers and they talk about one gene or another, if they're talking about the gene, it's in italics and lowercase. If they're talking about the protein, it's capitalized. And here's the reason for that. If this is the virion here, when it actually reverse transcribes, makes a DNA copy, it has to integrate that copy into the host genome. This is what that sequence looks like, called the provirus. And the provirus now has this identical sequence over here, the R and the U5 and the U3 and the R at the 3' end. But now this part is now also over here. This part is also over here. In the process of reverse transcription, the regulatory regions are duplicated at either end. This is now called a long terminal repeat, and it has the promoter elements where they belong, still has the poly A elements, and they're silent in the actual transcription. And here at the 3' end, we have the poly A sequence, and the genes can be expressed appropriately. So this is just a glossary of terms, and for those of you who don't have to deal with this on a daily basis, but maybe have to read these papers on a daily basis, you can you can always tell a virus with a program. This is what the virion. This is a cartoon of the virion. It's enveloped that it gets on its way out of the infected cell. On the surface, it has these glycoprotein spikes, which are going to be involved in attachment and fusion into the next cell it tries to infect. On the inside, the guts of it are these 2 copies of RNA. Now, these are not complementary. These are 2 identical copies, plus stranded, same direction as the direction of transcription. So 2 copies of that, plus the enzymes, right? And it carries with it its own primer to prime synthesis when it gets into the next cell. And that primer turns out to be a tRNA coding for lysine. It stole it out of the cell that it had just been in, and that's what it uses as a primer. It contains reverse transcriptase and also the integrase gene product that it'll need to integrate its genome. Moving on, so just to go through the replication cycle of HIV, and like Google, I've tried to get out of all of those to get my sans serif fonts in order, and I've missed a few. But the first steps, and I want to point out that this has a lot of arrows on it. And I'm not trying to make it complex. The reason it does is as all of these steps have been elucidated, they turn out to be potential sites for drug development. So every step in HIV replication essentially requires a virion gene product and a cooperation with the cell. It can do nothing without the cell, as you'll see. And each one of those cooperative interactions is either more virus or more cell. But the interaction itself is essential. The virion part of it represents an Achilles heel that a new drug can be developed. So it really is worth trying every, what sounds like, oh, that's just one more nerdy little step, turns out to be a very useful step in trying to identify places that the virus can be inhibited. So for instance, to get to the surface, to get inside the cell, you might imagine, oh, it just has to get stuck onto it and then stuff will happen and it'll get in. Well, that has actually been dissected out into 2 steps. So first you have to attach, and second you have to fuse. Both of those steps are distinct, and both of them have drugs that can inhibit them that are now FDA approved and used in our clinic here. So let's talk about those for just a second. If I can. Right. So attachment and entry, two distinct steps, requires viral factors. For attachment, it's the envelope glycoprotein that's sticking out on the surface. That's there. It's going to attach, looking for host cells that it can interact with. And the entry step, driving that virion through the membrane, is the envelope glycoprotein that's non-covalently linked to the GP120, and it's called GP41. Now, there are host cell factors, right? To attach to the cell, we need a surface marker called CD4, but that alone is not enough. A second surface protein called a co-receptor is also essential. So just having CD4 on the surface is not enough. We need either one of these co-receptors, CXCR4 or CCR5, and they're commonly found in a number of different cell types in the lymphoid lineage, but they're also found in other places as well, notably the central nervous system, where microglial cells will express both CCR5 largely and to some degree CXCR4. So, okay, well, if it needs to bind both CD4 and some co-receptor, why don't we develop drugs against that interaction? And the one that has been developed so far and is clinically used is a co-receptor blockade. And here are their structures. I'm sure you've already got them all memorized, but basically what it does is disrupt the interaction of the viral protein GP120 with the co-receptor itself. And because that co-receptor interaction is blocked, the virus can't enter. Even though it can engage CD4, because you're blocking the next interaction, it can't attach to the cell and interact. It's also possible to block the fusion event. So let's say attachment takes place anyway. Well, once GP120 attaches to this CD4 and its co-receptor, that causes a conformational change in GP41, and now GP41 has these 6 helix bundles, very much like, reorganized, very much like a spring-loaded mechanism. So there's a great deal of potential energy stored in these alpha helices that reorganize on the binding. And that's a key piece, because once that occurs, you have a lot of energy built into these GP41 molecules that can essentially push the virus through the cell membrane. So this is not one of those occasions where, perhaps when you were younger, or maybe now if you're young like me, and you blow bubbles and you see bubbles floating around and you see 2 of them sort of come together and then they pop and they become one. That's not what's going on here. You need a substantial amount of energy to push the virion membrane into the host cell membrane so that they fuse and the contents of the virion gets into the cytoplasm of the cell. And that energy is provided by the potential energy that's in this spring-loaded mechanism. All right, well, if that's clear, this is what some of those alpha helices look like. Terrific. Well, if that's specific and, it's dependent on a virion sequence, that might not be a bad place to develop a drug. And the drug that was developed is very much like a monkey wrench. It looks like an alpha helix, and it's in the opposite orientation, but it can bind to this and gum up the ability of that spring-loaded mechanism to actually transfer the potential energy that's in the alpha helix into kinetic energy, pushing that virion through. So this is for those of you who are interested in drug development, this was a tour de force. It's 36 amino acids. It's the largest peptide, I think still, made for clinical use. The company had to build their own building just to generate this peptide. Downside is it's a peptide, so it gets digested. If you take it as a pill, it has to be injected. And okay, let's suppose you get past those two steps and the virus actually does what it wanted to do and push the contents of the virion into the cytoplasm. The next step is called uncoating. And it's called uncoating because it is just so diffuse, nobody understands it really very clearly yet, except to realize that it's an absolutely essential step in virus replication. You might say, yeah, sure, you got to have access to nucleoside triphosphates. It's got to encode. It probably just falls apart. No, it is a highly specific event in virus replication. So much so that if we had done things just a little bit differently, and I'll show you how much differently we could have done things, we wouldn't have this problem now with HIV infection infecting 35 million people. So here's just to try and show it in a different kind of cartoon. The virus has fused with the cell membrane, and now this core structure containing these two RNA molecules, plus stranded RNA, plus the enzymes, are about to reverse transcribe. But this thing has to be made, has to uncoat in some fashion. And that uncoating actually takes some cooperation between the host and the cell. In this case, it's an antagonistic interaction. There's a host cell gene called TRIM5 alpha. TRIM5 alpha can block the uncoating step in HIV replication and also block the uncoating step in a lot of other viruses as well. So great, if we have this gene, come it's not working. And the reason is, it's good, but it's not that great. We got the wrong one for HIV. And I'll just show you how that looks, right? The virus protein that TRIM5 alpha recognizes is the GAG protein that forms the structure of that core that's protecting those two molecules of HIV RNA. And here's how bad things are. Everybody got a TRIM5 alpha. You do, I do, monkeys do, chimps do, and keep going back, and they all have a TRIM5 alpha gene. It makes a protein that's in the cytoplasm. It's one of those innate immune molecules that senses foreign molecules in the cytoplasm. And in this case, it's a viral gene product. Well, with HIV, it doesn't work so good. And I'm so sorry. Go back. Here we go. With HIV, it doesn't work as well, and HIV can infect that cell. All right. Well, what about chimps? They got a host TRIM5 alpha. Can HIV infect their cells? Yeah. They got a crummy one too. How about monkeys? They're not that far away, especially from evolutionary, especially from people in my family. So do they have a TRIM5 alpha? Yeah. Can HIV infect those cells? No. And it stops at that step. So you can attach, you can get that core in, but that core does not undergo reverse transcription, and that's because this TRIM5 alpha blocks it. All right, so they got something we don't have. Let's look at a different virus. That different virus is in the chimp family, and it's a virus that's just like HIV, called an SIV chimpanzee version. Can it infect humans? Yeah, probably did. That's how we got this problem. Can it infect chimps? Yes. How about the monkey? Not really well at all. So this monkey TRIM5, and again, the restriction is right at that time, at that point. How about the SIV, right? Now, SIV can infect monkeys. Let's face it. It's simian immunodeficiency virus. That's where it comes from. It infects real well. It also infects, it can also infect humans, and chimps not so well. So if you're scoring at home, we didn't do so well on this TRIM5 alpha test, and monkeys did that much better, okay? And it's been blocked at that particular spot. And this is what's going to gall somebody. If we had any sense at all, we'd have picked the monkey TRIM5 alpha, for crying out loud. Why didn't we do that? And I'll explain why, but the difference is are really minor. So you can model, obviously, you can model this whole infection system in tissue culture. And you can make the human cells have the monkey TRIM5 alpha. And when you do that, the human cells are not infectable with HIV. You can protect human cells with a monkey TRIM5 alpha. Doesn't that just the gallingest thing? I mean, we can do all this molecular biology. Well, I don't see those monkeys doing any molecular biology. How come they get to be so lucky and we don't? And it turns out that TRIM5 alpha is going to restrict other viral infections as well. And as you probably know, retroviruses have been infecting us, chimps, monkeys, and vertebrates for millions and millions and millions of years. And if you look at these genes and compare them among species, they are some of the fastest evolving genes in the genome. In other words, they're responding to a selective pressure. And it's thought that the selective pressure are waves and waves of retroviral infections and other infections that happen to jump into our species. And so, you know, there's this old expression, generals are always fighting the last war. The TRIM5 alpha that I've been complaining is a nice word, complaining about for the last 5 minutes are probably the one that saved the species the last time we had a problem like this. As if there are plenty of TRIM5 alphas and we're getting slaughtered left and right, and the one that grows out is the one that can prevent uncoating of a viral infection in a wave of retrovirus, some other virus, that is selecting TRIM5 alphas very, very quickly. So it's a theory. It's not bad. Sounds pretty cool. But you know, between you and me, I'd rather be studying this as an oncogenic virus and not have it as a human immunodeficiency virus. But this 1 or 2 amino acids are why you guys are subjected to this lecture this afternoon. So evolution can solve the problem we have with HIV, but it's going to take too long. So if it goes to 35 million, we'll lose a lot more before the variant grows out on the planet that can resist this. So we've decided to go in a different direction to try and deal with this virus infection. All right, so that uncoating step, as vague as it is right now, because it's not well understood, that might not be a bad place to develop a drug, and it's undergoing a lot of drug discovery. But this is a tough one because it's not binding to a specific area of that GAG molecule. It looks like it's recognizing a much larger structure. So stay tuned, but that looked like a potent place where we could get a very useful drug. The next step, once this uncoating occurs, is reverse transcription. Now this one is one we know a little bit about because that's an enzyme. And if you'll notice, some of our best drugs are enzyme inhibitors. And one of the reasons is because they can employ folks like us to do very high throughput assays when you have an enzyme assay that actually you can screen an awful lot of drugs. Uncoating, I don't know how to screen for that yet, but I know how to screen for an enzyme. And so those were the first drugs that were developed for HIV. And just to summarize, reverse transcriptase, this enzyme that's going to take those 2 molecules of RNA and make a cDNA of complementary 2 strands of DNA, is an RNA-dependent DNA polymerase. That makes sense, right? Depends on RNAs to make DNA. Well, once it's got that RNA-DNA hybrid, as some of you probably know if you've worked with RNA, that an RNA-DNA hybrid is actually quite stable. And so you have to chew out the RNA to get the next strand actually made. And the act of doing that is done by another function of reverse transcriptase called RNase H. And I say this because it digests almost all of it and leaves a couple of little areas of RNA DNA, and they become the primers for the next round of synthesis. This is great. It's doing it all by themselves. And then it's a DNA-dependent DNA polymerase. So it takes that DNA that it just made, those little RNA primers, and makes another copy of DNA. You know something really irksome about this virus? This is an RNA virus, and it has its own enzyme, and it can't make any RNA. It can only make DNA. It can make DNA out of RNA. It can make DNA out of DNA. It can't make RNA. It's going to make RNA in the cell, and that's what's going to get incorporated into the virion. Anything and this is good. I don't know if any of you are interested in viruses or RNA replicating enzymes in any kind of way. Anything that's got to deal with RNA makes mistakes a lot at a much greater frequency than things that have to deal with a DNA template. If you're at a DNA template, you can go to the 10 to the minus 10th and not make any, the error rate is 10 to the minus 10th per site per cycle. You start dealing with RNA, and it goes down by 4, 5, 6 logs. So with HIV and all retroviruses, it makes mistakes on the order of 1 to 4 per every 100,000 bases synthesized. Now you think that's, look, I wish I made mistakes in that frequency, right? Every 100,000 things I had to do, I'd like to make just one mistake. But this is still 10 to the 5th fold less faithful than the DNA replicating enzymes. In other words, every round of replication, if you have something that's 10,000 roughly 10,000 nucleotides in length, which is what HIV is, and you're going to make, it'll be 100,000 bases before you make the first error or so. You go 10,000 this way and 10,000 coming back, that's 20,000 bases. Essentially, every molecule that you synthesize has at least one change in it. Some of those changes are silent and they won't matter. Some of those changes, a lot of them, are lethal. So replication is terribly error prone, and it's rapid. So HIV can replicate, the replication time is something like a day. And that means that before I ever start antiretroviral therapy in our patients in the clinic, there are likely to be single nucleotide mutations at every place in the genome that can tolerate a change. In other words, single nucleotide changes that confer drug resistance pre-exist prior to starting antiretroviral therapy. And all I do with a crummy drug is select the ones that can grow in the presence there. So I'm not making mutations with these drugs, I'm just selecting them. And I think what I want to argue is that this isn't just an old virus that doesn't know how to make nucleic acid. I think, and other people think too, that this is a pathogenic determinant for virus persistence. And so here's an example of that. Let's take, and I know I know you guys know the genetic code. I know you got the thing memorized cold, right? And you know that CAG, that codon, codes for glutamine. And let's just say we're in a position 151 in the reverse transcriptase molecule. It's usually glutamine, right? CAG. But sometimes it makes CTG and you get leucine instead, or AAG and you get lysine instead. And if you make two changes, you can get to ATG, and that's a methionine. And all three of these can occur. They exist probably in a very, very low frequency, but they're present. 1 in 10,000, 1 in 1,000, something like that. This one is very, very sensitive to all the nucleoside inhibitors we have for HIV. And so are these two. This one is resistant to all of them. In other words, making two changes makes you completely resistant to a whole class of drugs that we have to inhibit it. All four of these are probably pre-existing prior to starting the antiretroviral therapy. So one change that could cause big problems for therapy is already there. Now, obviously, the virus isn't doing this because it knows we're going to invent drugs, but this is the same kind of mechanism that's been invoked for the virus to evade an immune system that can't keep up with all of the diversity. So if you have it's just like a society. If you have a society that tolerates its fringes, you can do a whole lot more than one that's only monomorphic. So the virus population can respond rapidly to some selective pressure. And in general, that's going to be an immunologic one. For us, it could be an immunologic one or it could be a viral one. I don't know how many of you are interested in or study immunology. The development of CTLs is just not fast enough to keep up with single nucleotide changes. And that's why when we develop drugs, we can't just use one. We have to use enough drugs with different mechanisms of action, or really different resistance profiles to make sure that we have at least 3 so that resistant can't develop as quickly. So that's reverse transcription. Once the reverse transcript is made, it has to get transported to the nucleus, integrated into the host genome, and that's done by the host by the viral gene integrase. And now we have new drugs available in the last 6 or 7 years that turn out to be the best set of drugs in the past 15. Integrase inhibitors have really revolutionized the way we've been able to deal with viruses that have become resistant to that first round. And so they're not that far off what Yves was talking about in the last lecture. In fact, he developed some of the first inhibitors for integrase. They were relatives of these anti-cancer agents. And it turns out that they were good, but there were better ones to be made when when they developed, and you know, he had a piece of all of this action, when they developed new assays to dissect apart the integration reaction and develop drugs that could inhibit that set of reactions. Once it's integrated, the enemy is within, and it produces RNA, as I said, using cellular enzymes. So this is RNA Pol2, which transcribes the HIV genome. You can imagine once it's integrated in the genome, it's only 9,000 nucleotides, and the host genome is a lot more than that. The promoters look very much like cellular promoters. It just makes RNA as if it didn't know what it was doing. It has to transport that RNA out into the cytoplasm. It's translated on ribosomes, assembled at the cell membrane using the escort, co-opting the escort pathway, and then the virions have to assemble at the cell membrane and then undergo maturation. And that particle maturation is a really interesting step, and that was really the genesis of a different set of drugs called protease inhibitors. As you can see, the virus, and I don't know how well you, how familiar you are with electron microscopy, maybe something. Here's the cell, and here are the virions budding from the cell. And those of you who are familiar with electron microscopy know that it's just like any other microscope. We're going to pass a light source through a sample, and then we're going to take a reading by looking through it, right? And with a light microscope, you know how that works. You stain it so you can see what's in there, because the cellular material, it's sort of, you know, pretty amorphous, and it's hard to tell apart one structure from another. So you got to stain it specially so you can see stuff. And then you can look through a light microscope. The light comes up and you can see one thing or another. Electron microscope is the same thing, only you can't look into it. You have to make a recording of the electron beam coming through it. And you got to stain it too, because it's the same problem. So the stains for electron microscopy are really sort of interesting. Anybody know what they are? Osmium tetroxide, right? Lead. Now, those are 2 of them, and you guys know these guys did really well in chemistry, right, Terry? These guys, that's how they got here in the first place. You know that both osmium and lead are very highly positively charged molecules, right? And you can just imagine if you're staining something with a positively charged molecule, it's going to bind the thing that's negatively charged, right? So this is the easy part about interpreting electron micrographs. All you do is look up what they stain it with, and as long as it was a positively charged molecule, you're not looking at cellular structures. You're looking at the heavy molecules that are deflecting the electron beam as it's coming through the sample. And so all I'm really looking at here is osmium. And where'd the osmium go? Well, the osmium usually goes to the place where there are the most negative charges, because it's stained in a way that you facilitate charged interactions. So I'm looking at where the positive charges, the stain is, but I'm really looking at the negative charge. Now, what's got the most negative charges in a biological system? Well, you got your nucleic acids, right? They've got a negative, they got 2 negative charges on every base as it's going through there from the phosphate. That's what I'm looking at here. So all I'm staring at on this side over here, where this is all really dark, where all the osmium or all the lead went, that's where the nucleic acid is. Well, over here, it's plastered up the inside of that spherical virion as it's coming out, along with the gag protein. But mostly, that's where the RNA is. Over here, the RNA is condensed in that core that's going to be forced into the next virion. So this is a really big shift. And I'm making a big deal out of it because it needs a viral protein to mediate this, which is called an immature particle. And I put a bunch of them there so they're easy to remember because they're all immature. They're in a corner all together giggling. But then once they're treated by protease and they become mature and they can stand on their own. But this process occurs as budding is taking place and after budding is taking place. In other words, I can get victory from the jaws of defeat here with a protease inhibitor because I can block this reaction occurring. These virions, they got everything in them that this one has. It's just in a different conformation. This one's infectious and this one isn't. And so protease inhibitors were developed as an inhibitor, just like enzyme inhibitors, and they turned out to be the best thing early on to be used in conjunction with RT inhibitors to save people's lives, essentially, because that's the genesis of combination antiretroviral therapy, right? And it was either two nucleoside inhibitors and a non-nucleoside inhibitor that inhibits reverse transcriptase, or these guys and two nukes. Got to use them in combination, otherwise you'll get drug resistance immediately. But this is what changed the epidemiologic curve of HIV infection. So now that we've made some viruses, let's see what they do in human populations. As I said, we've looked at this you can see both HIV-1 and HIV-2, and again, HIV-1's closest relative is in the chimp. HIV-2's closest relative is in the sooty mangabey. And that implies directly how these viruses may have interacted with humans. But before I go there, I guess I put this one in first because, this is our commercial break for neoplastic diseases. HTLV-1. I don't think I put it down here. So have you heard of HTLV-1 before? Human T-cell leukemia virus, discovered here, Robert Gallo, and in Japan. Japan, really? They have it in many, many cases of this of HTLV as well. How many? So show hands. How many people have heard of HTLV before? How many people haven't heard of HTLV before? How many people don't have hands? Never mind. Okay, fine. It was a joke. How many people do you think in the world have HTLV? Yes. Oh, this is virology, right? So we do everything in log terms. Is it a million? No, let's go 100,000, million, 10 million, 100 million, a billion. A million. I interpret that grimace as a million. Anything else? What's that? 100,000. 100,000 going once, going twice. Anything more? 20 million. There are almost as many people infected with HTLV in the world as they are with HIV. What? Don't they have as good a press agent? I mean, what's the deal here? What's really interesting is that of those 20 million, it's only about 8% of them that get disease from the virus. So 20 million are infected, 5% get leukemia, 5% get another disease called ham-TSP. There's another very close relative this time of HTLV-1. You might imagine what it might be called, HTLV-2. HTLV-2 is in a lot of individuals as well, and it's transmitted slightly differently. It doesn't cause any disease at all. So here's a disease of humans, transmitted. It's a virus, HTLV-1. Here's another virus of humans, but it doesn't cause any disease. I don't know if it ever did or not, but it's endemic in places like Amerindian populations in South America, in Central Africa. There's a lot we don't know about humans that we can learn by studying these viruses. But HTLV1 is highly endemic in places like Okinawa or in southern Japan and in the Caribbean, where it can cause significant disease only because there are so many people who are infected. It's transmitted by breastfeeding. So if you block breastfeeding, you can actually reduce transmission about 80 to 90%. And the diseases it causes are an acute T-cell leukemia, which can have a long latency. So you get infected by breastfeeding, and about 30, 40 years later, you actually start to develop the disease in a relatively small number of individuals. It has a number of immune deficient associated diseases with it. And there's chemotherapy, some of it being developed here at NIH. This one's a tough one because it's the cells that are expanding. And so they've already gotten infected. Attachment inhibitors, it's not going to work. The cell's going to get transformed. It doesn't matter. Reverse transcriptase inhibitors, only way back in the beginning could they possibly have helped you, right? And then integrase inhibitors, no, enemy's already within. So the therapy isn't antiretrovirals, it's therapy against the cells themselves. Okay, so let's get back to HIV now, about 35 million. It's probably tops HTLV1, but they're in the same order of magnitude for sure. Where did it come from? And so the initial inferences came from that phylogenetic tree. Well, if there's a virus that looks just like HIV in the chimp population, maybe we ought to go look into that, right? Or if there's a virus that's just like it in the sooty mangabey population, maybe we ought to check those animals. And so I know you guys have hard lives. The postdocs of Beatrice Hahn, they got hard lives too, because they went and they sampled chimp populations in different parts of Central and West Africa looking for HIV-like viruses. And some of you are smiling because maybe you know how this goes, because it's not like people. You can't get informed consent from a chimp. You're not allowed to bleed them. So, all right, that's going to make this a little bit more difficult. What are we going to do? Wait for them to die? We can't do that. Well, as it turns out, you can get their urine and you can get their poop. And from those things, maybe you can get some sequences. And that's exactly what these postdocs did. They collected, and these are reserves where the animals are protected rightly. And they said, listen, we know all about them. We got them named. I can tell you where they sleep at night. And you can go pick up their urine and whatever else you want in the morning, as long as you don't disturb them. And so they did. And they also, and when they were looking at PCR HIV or HIV-like sequences out of them, they were also PCRing their HLA types to make sure that this was really chimp poop. So great job, right? Turns out that all these populations here in Central and West Africa had HIV-like viruses in them. And so they were named simian, because they're not human, simian immunodeficiency virus that came from chimps, CPZ. And it turns out that there's another group of chimps that are over here in a different part of West Africa. They don't have any of these viruses, for reasons that remain poorly understood. They got there, they think, about 10,000 years ago. So whatever happened probably happened to these guys, probably from the monkeys in the last 10,000 years or so. Or if these guys got it, the ones that got it died and they had a breed that lived. They can be infected. There's no problem infecting their cells. You can't do that anymore either. But there's no problem infecting their cells with HIV. It just happens that these populations don't have any. And so the theory, and again, it's just a theory, is that much of this may have started with people like poachers or hunters. Let's face it, primates have been eating primates in all parts of the world ever since they started to diverge from each other. So this is not a new event. And I think the things that drove it may have been new, and we'll come to those. But it's thought that there's some interaction between the animal kingdom and the human, be it poaching or something that allows for transmission of blood and body fluids. And you can imagine that this is something of a violent act. The places where these animals are then taken is called a chop house. You can also imagine that butchering these things gives you ample opportunity to transmit bloodborne viruses. And then they're sold in bushmeat markets. I don't know if any of you have been to Africa. I haven't, but I've talked to plenty of people who say these are thriving places for people to get protein in Africa. So, there are at least a number of places where viruses can be transmitted by blood and body fluid. Now, if you look at the HIV sequences that are in the humans, one thing you'll notice if you use very specific phylogenetic techniques is that you can tell them apart by their phylogeny. And it looks like there are at least 8 or 9, maybe even 10 different variants. Are they so different that 10 different times things jumped out into the human population? Perhaps. Some of them seem more related to one another, and perhaps they've grown out. Anybody do any of this phylogenetics or Bayesian analyses of sequences? You can use it to identify common ancestors using coalescent theory. It's the theory of Eve that everybody can be traced back by coalescent events to a single set of ancestors. And using that analysis, you can trace back HIV at least close to 100 and maybe more like 130 years. So this isn't a virus that jumped into humans in the 1960s and 1970s. This is something that has been in human populations probably for a whole lot longer, likely close to 100 years. Well, like, how come, right? How come we discovered it in 1981? Or how come it got to be an epidemic in 1981? What is the deal? And the deal is that the spread of the virus, it's sort of, I think the analogy that people have used, it's like starting a forest fire, right? Not every spark produces a forest fire. It's likely that these events, zoonotic events from the animal kingdom into us, were probably occurring all the time, but they were of such low frequency and they weren't transmitted among large populations that one or a few just died out. So we never had that opportunity to evolve a TRIM5 alpha that could resist this virus. So yeah, there are the reasons why you could get the transmission, but then there are these non-biologic reasons, political reasons, economic reasons, and multiple epidemics that allowed the virus to be transmitted in the modern age. And so I just want to highlight a couple of these in the last few minutes here. Here's a map of Africa, and it's clear that the oldest viruses are in Africa, and this is where they arose from. Here's a map of Africa in, I think this is 1812. Let me see if I have it here. 1812, the late 1800s, and then 1960 when 1980, when a lot of these countries got their independence. And I think one thing you can tell is during this colonial period and post-colonial period, there was a great deal of change. And this change was characterized by large population upheavals and by a significant amount of malnutrition. Malnutrition itself is an immunodeficiency condition for humans. So that puts them at greater risk for infection with viruses. And this movement of large populations over distances may have facilitated infection. Population growth. So here is the population, the human population in Africa during the time that we're thinking about, last 100 years or so. And so again, you know, we're virologists here. You're virologists for a day. So this is a log scale here, and this is years from 1910 up to 2000. And you can see in that early period, just a couple of million people were living in all of Africa. But that expanded greatly during the period when this virus was probably taking hold. And in fact, it may have come along with that rapid expansion. So here, you go from 1 million to over 10 million in less than 20 years. We go to like 20 million. Do we get to 200 million? Not for another 30. And we still haven't gotten to 2,000 million. So this period in here was a time of great population expansion. And if you trace back the earliest viruses, they seem to be present in places where this population expansion took place. Kinshasa, in Central African Republic. There's a place where they have pictures from the 1910s and 20s, and it's very, very rural. And then within 30 or 40 years, it gets much more urbanized. The ability to go around improved greatly in the 20th century. This is what's called the Trans-Africa Highway. I know when I first heard about the Trans-Africa Highway, I thought it's like 270. But it's not. It's just this road, but it's punctuated with places to stop and eat and obviously engage in other economic activities that viruses can be transmitted therein. So this highway this highway has cut through Africa, allowing the goods from the center of Africa to get to both coasts and to allow other things to spread as well. All right, so I think that helps explain perhaps why things happened in the 20th century. I think these things the zoonotic events have been happening a lot, but they've been accelerated in the last 100 years or so. How about us? How come we got so lucky? Where did this virus come from? And I don't know if any of you have ever read Randy Schultz's book, And the Band Played On. There's this urban legend, and urban legends are just the best, that the virus came to the United States through a flight attendant, and his name was Gaetan Dugas. And Gaetan Dugas was very, very popular. And after he got infected and obviously had Kaposi sarcoma, he said, I'm the first one, I brought it over. But, behind every good urban legend are people like you guys. And I'm just fine. I see your faces. I know you're nerds. Nerds took that sequence analysis thing to heart, collected sequences from everywhere they could find them in the Western Hemisphere and compared them to those that were in Africa. And this is work of Mike Worobi, and found that the earliest viruses in the Western Hemisphere turned out not to be in the United States, but turned out to be in Haiti. And so there was a great deal of interaction between Haiti and Central Africa in the 50s and 60s, mostly 60s. And it's thought that their advisors may have brought the virus back there. And from there, it spread to other islands in the Caribbean and to the United States. And so you can't find viruses in any of these countries that's older than the ones in Haiti. So it's likely that it made the first jump to Haiti and the second then to the United States. And at that point, things were just completely out of hand. This is the growth of HIV cases in the United States. And once again, we're all virologists here. This is in February of 1983 when there were about 1,000 cases depicted by these orange dots in highly urban places. This is Chicago, New York, Miami, Houston, San Francisco, and Los Angeles. And now just see how this grows. So in 2 years, 1 log, okay, 10,000 cases. In 4 years, another log, 100,000 cases. And now, 6 years later, we got half a log. We never got the next log. So most of the epidemic occurred in those first 5 or 6 years. Here we are at 500,000. And now, as you guys mentioned, we're only up about a million or so in the country. It's stabilizing as an epidemic over the last 20 years. Currently, who gets infected? 53% of all new infections are still men who have sex with men. Heterosexual, 31% IV drug use. Here, cut off about 8%. and here women are somewhere around are getting close to 25%. African Americans exceed all other groups in numbers of cases of HIV. Those first maps that we saw, highly urbanized, right? But now 12% of cases are in populations with less than 50,000 individuals. It's spreading out into communities. As I mentioned, 25% of all AIDS cases are now in women, and about 50% of people living in the United States now with HIV are over the age of 50. So this is no longer young gay men. This is an endemic that persists thanks to antiretroviral therapy. So let's talk just briefly about therapy. This is a sign that was on a bus when I think when I first came to NIH over 25 years ago, where if you get the AIDS virus now, you and your license could expire at about the same time. There was no therapy. In the last 30 years or so, this is now a timeline of all the antivirals that have been developed. First AZT here in Building 10, DDI, DDC developed also in collaboration with the NIH, as well as D4T. And then each one is color-coded as according to how well or what target they have. And you can see the development of these protease inhibitors really changed the world. And now new drugs coming on all the time. The pipeline, though, I must say, is dripping. And so new targets and new drugs are slowly coming on board. And the idea is to try and stay ahead of drug resistance, right? This is something called the Gardner Continuum of Care that really explains the epidemic in a very simple slide. So, well, how many people, we figure there are about 1.2 million people in the United States living with HIV. How many of them we know about? How many have been diagnosed? About 86%. How many of those are engaged in care? 40%. We're missing a lot. How many of those who are engaged in care are actually getting antiretroviral therapy? 37% of this original 1.2 million. And how many of those have their viral RNA levels suppressed by that antiretroviral therapy so that they're not developing drug resistance and they're not progressing in their disease? And so it's 30%. So this is the number, this is the proportion of the epidemic that's actually reaping the true benefit of antiretroviral therapy. The difference here is a group of individuals that are developing drug resistance, which they can transmit. And once drug resistance emerges, new regimens are essential. We got them, they're useful, but they're not perfect. We can run out. So there's a real issue there. The key advances now in HIV therapy in the last 5 or 6 years, pre-exposure prophylaxis. Let's give the drugs to people who are at high risk and not to the people, and also to the people who already have the disease. This way we can block transmission. That's been demonstrated to work if individuals are adherent. There was something called the SMART study that demonstrated that continuous therapy was essential to avoid AIDS complications. So just because you were doing well, we could stay off therapy until your CD4 count came down and you had an opportunity. That turns out to be not the case. You need continuous care. And then recently another study saying the earlier you start, the better off you're going to be, the fewer, the less of an immune loss you take. In other words, diagnosis equals engagement in care and starting therapy. If you don't do that, we're going to have progression of disease. That becomes a much more clear-cut thing to do and a much more expensive one around the world. So the next advances in HIV therapy is things you might imagine, vaccines that people have been working on forever, and I think making stepwise advances, but no big blasts, and cures. Why don't we just cure this stinking disease? HIV-infected cells persist for the lifetime of the patient. It's going to be tough to get rid of them, but that's a big research, part of the research agenda in the HIV community now, and this is something that our group is working on as well. There's one person who's been cured. His name is Timothy Brown. He had a bone marrow transplant for a comorbid leukemia, and that's about it. And he got very, very lucky. I'm sure this is the person you heard about in the news, but that's pretty much it right now. So I know I've probably gone over time, but and I'll stop there. But the lessons that I think we've accumulated over the past 35 years are that viruses are bad and should be avoided, except when they save the planet, like reverse transcriptase did, and maybe if it saves you from the next virus, the way TRIM5 alpha was educated last time, could be the way it could be done again. Epidemics are really not single events, and they evolve. We have an endemic HIV infection now. Antivirals are useful, instituted as early as possible. Adherence is essential. So I'll stop there and see if we have any questions.
Can you clarify in the prior slide you said this was for the ones that are encouraging the development of resistance to therapy? Are you talking about people who are on the therapy or people who are?
Yes. So they've been prescribed antiretroviral medicines, but they're not fully suppressed. In other words, they have virus present that's growing, despite the fact that they're on antiretroviral therapy. And so when non-adherence occurs, then it's like you only have a little bit of the drug around. That's a great way to select for drug resistance. And those drug-resistant strains can be spread in the community. So in any given urban area where drugs have been available for as long as drugs have been available, you can find that 10 to 15% of newly diagnosed individuals have drug resistance mutations, requiring a different regimen for to be used, and sometimes they have to be much more complex. In the District of Columbia, we've been collaborating with a group in Georgetown, and the data look, I hate to say it, look a little bit worse. And there's more like 20% of individuals have drug resistance, and those individuals have been around for a prolonged period, as early as these drugs have been around for. So in the middle 1990s, stored samples have suggested that there was a considerable amount of drug resistance present. Okay, so that's something I didn't get into because I'm a lazy bum. HIV-2 is a virus that is very closely related to a virus that's in sooty mangabeys. So this came from, we got one from a chimp to humans. This is one from a monkey to humans. And those two viruses are close to 98% identical. That's where one came from the other one, no question, right? But the virus doesn't grow as well in humans. And so the disease is slower than it is with HIV-1. So patients usually get diagnosed in HIV-2 exclusively infected. They're usually mostly in West Africa, except when they move here and we diagnose them, right? They get diagnosed older because they don't progress to AIDS as quickly. Now, initially, people thought, oh man, maybe we can do something with this HIV-2, we can make a vaccine out of it because it's slowly growing and maybe it'll prevent you from getting HIV-1. And so as they were doing all these wonderful epidemiology studies in West Africa, picking out who was infected with 1 and who was infected in 2, they basically, made maps of where HIV-2 was, terrific, and how many people were there. They went back 5 years later and they found the very same people who were HIV-2 infected were now infected with HIV-1. So it didn't prevent their infection. And that's been the common observation, that it's not a vaccine, and you can be infected with both. And it's the HIV-1 rate that takes over. So HIV-2 becomes a little bit more of a bystander. But you can be infected with both. HIV-2 is a fascinating disease because it is much more like a monkey virus, and that monkey virus is intrinsically resistant to some of the drugs that we normally use. So for our fellows, one of the things you just pound into them is that if you see somebody who's from West Africa, even if they're on medications, even if they've been, in the system for years, and we test them for HIV-2 to make sure that we're not missing something. because we could be using the right regimen for HIV-1 and the wrong one for HIV-2. And unless you think about it, and in West Africa, they always test for both.
You mentioned that microglial cells also have the same co-receptors.
You guys remember everything. Just that was the beginning of the lecture. Can't you forget it?
So how does the HIV actually bind to microglial cells then?
It infects them.
It affects them. How does that affect patients then?
It could be bad. That's a really good question, and it's the subject of a lot of intense interest on a couple of levels. First of all, how does it affect the brain? Second of all, do these drugs get into the brain, and can they suppress virus there? And if they don't suppress it as well, is that a sanctuary site in people who are suppressed for new viruses to evolve that are resistant to the little drug that gets in? So that's an active area of interest for a lot of places. And basically what people have found so far is that some of that the viruses are generally suppressed in both places. So we get enough drug. I know this is an area that people are going to discuss because the drug levels are lower, but the drug levels are always lower and protein binding is different. The virus is suppressed in the brain, but soon after infection. So people have done wonderful studies of recently infected individuals. You could find the virus in the brain. So it spreads there very quickly. Some of the viruses that circulate in the brain have different genetic signatures than the ones out in the periphery. But these are sequence differences, not drug sensitivity differences. They grow mostly in the microglial cells. And you sound like you know how to spell microglial. So these things they're not neurons, but they're supporting neural function. You lose enough of those and people get encephalopathic and they get what people call HIV encephalitis. And so thinking is highly impaired. All right, they start on antiretroviral therapy and the virus in the periphery is reduced and the virus in the central nervous system is also reduced. And life is good, right? No. And so after long-term therapy, there's something called HIV-associated neurologic dysfunction, or HAND. And HAND looks like it's a cognitive dysfunction of infected individuals that's present even after they go on antiretroviral therapy. And it's not clear whether it's progressing on therapy because there's a persistent immune activation after the virus has been suppressed. There's still antigen around. You can always tell there's virus present because there's an immune response, or something else, or whether it depends on how bad things were before you started therapy. But it's an area of active interest. And, if you're interested, there's a group in the NINDS, Avi Nath, the clinical director, who's made real strides in trying to understand both the HIV effect on the brain itself and then after suppression. So yes, yes, and yes to all of your questions. Okay. Yes, I quit. But yeah. Oh, that's because I can't shut up. He takes the microphone. I just keep talking until I get another email to come back, and it's just like. But I really, really do appreciate you staying such a.